So light therapy is kind of holistic, it's natural, there's no side effect. It kind of get a brain to do its work. It does not do the work on its own.
What you have uncovered is, I mean, the single best term I could describe it as profound. This simple application of light can have such a radical improvement on a disease that's going to devastate our society. There is a tsunami of Alzheimer's disease approaching us, and there are no effective drugs. So what can you do? Hi, this is Dr. Mercola, helping you take control of your health. And today we're going to explore a simple strategy that can radically reduce your risk and probably serve to effectively treat many cases of Alzheimer's disease. So today we have on our interview Dr. Lou Lim, who is a clinician up in Canada who is expert in the use of near infrared therapy to treat Alzheimer's disease. And the extension of that, of course, is to prevent it. So welcome and thank you for joining us, Dr. Lim.
Well, thank you for inviting me. Well, let me first say that I speak with some evidence. We have just been accepted for publishing into a scientific journal. Our recent case series report is a clinical study on a small number of people with Alzheimer's disease. And that is quite, the findings are actually quite significant. Now, without going to the details now, we do get questions from skeptics like, how can light actually reach the brain to begin with and then get a response from the brain? Mm-hmm. So what I like to do is to actually try and demonstrate that first.
Okay.
And then you can see that this is not, all hocus pocus. It's actually doing something to the brain. Then we'll talk about the evidence.
Okay, we'll talk about the evidence and the mechanism and how this all works, because and there's a lot of associated details to this, and like most areas, the devil's in the details. So why don't you proceed with the demonstration and show us how light can actually penetrate the skull and have influence on brain activity?
Okay, so I want to introduce Reza Zomorodi. He's going to take over the interview from me. Now, Reza is one of the top neurophysiologists in Canada. He's attached to the Center for Addiction and Mental Health at the University of Toronto. So we're now actually working on more developments. We're at the cutting edge of getting images of the brain so that we understand better why some people are responding better than others. Say, even in Alzheimer's, we are noticing that some people respond virtually immediately and some are responding a little bit later. So that is, giving us more detail, and hopefully in the future we can actually customize intervention. We're really going to customize medicine here. So just to show you what it does, let me just pass over to Reza.
Yeah, hi, I'm Reza. I'm practicing neurophysiology at the Center of Addiction and Mental Health, University of Toronto. I have many years experience with the EEG and recording neurophysiology. So this device really got my attention. And for myself also was a very big surprise to see how the light could change the EEG and the brain signals. Because brain signals, or the language of the cells inside the brain, we believe it is the electrochemical signals, and we can detect the electrical activity on the surface of the scalp. And as a result, we expected to interface with the brain with the electrical or magnetic stimulation. And the light stimulation or photomodulation was kind of the surprising innovation for me. And fortunately here we have the device. We have a really high quality device, and we can record in high level close to the clinical standard EEG. And explore the effect of the photostimulation on the brain through the EEG and the change of the power spectrum or the frequency. And this is the settings we have here. We have an EEG with 19 electrodes with 1020 montage. This is very standard montage, so we have the location of the electrodes according to the standard. And we put the device on the surface and with the different location that we are stimulating.
And the device is the photostimulation approach that you're using to measure the impact on the changes in the neurological activity.
Exactly, this is the goal. This is, we want to see it here in the live, to see when we turn on the machines, how the brain responds to these machines. So it is the acute and immediate effect. But the mechanism, probably later Dr. will explain for you, but we believe it is kind of the interacting with the ATP, with the mitochondria activity, and it boosts the energy of the brain, and the brain uses this energy to generate more frequency, more oscillation, and organize or coordinate different network. Mm-hmm. And if you let me, I can start showing some.
Sure, go ahead.
Demo. Okay, so in the background, what you see, it is the electroencephalography signals from 19 electrodes. And I will ask Glenn to close your eyes. When we are closing the eyes, the main frequency happens. This is very well known. It is the alpha frequency. And we can see the alpha frequency very clearly in the data, and also the location of the brain with the activity. I'm going to show you where is the alpha activity inside the brain. This is a live demo of the brain activity in the alpha frequency. And most of the time you see the green light. Green light means the activity is a normal range. And if it is showing the red, it means it's the higher than normal, and the blue, it's less than normal. This is kind of the standard Z-score activity. And we are recording about 5 or 10 minutes of the EEG record, of the rest EEG, to have the baseline activity of the brain. Then we will turn on the machines, switch on the photostimulation, and we will see how this frequency is changing. Brain frequency, we define it in 5 different frequency bands, but they are not really separated. They are always interacted. And maybe the alpha is interacting with theta, maybe interacting with the gamma. They have different origin and different function, but at the same time, they coordinate together to process the information. And but for the rest EEG, when the subject sitting comfortably on the chairs and not processing any cognitive task, the alpha is a dominant frequency. That's why we are concentrating mostly on the alpha, because this is the biggest frequency, and right now brain is in the alpha state. And for the oral research, we usually record at 10 minutes, then we compare when the machine is on, and then after that, what is the effect of the machine on the brain. We can ask Glenn to turn on the machines, and we will see the effect on the brain.
Well, the machine, just for clarification, the instrument he's turning on is the photostimulation device that's allowing near-infrared radiation to penetrate through the skull into the brain.
That's great. So I'm not sure how the picture is clear for you, but you can see this green immediately went to the red. Mm-hmm. So it means higher activity, and really the machine is transferring energy to the brain.
Now, normally red is viewed as an alarm danger signal, and what does it indicate in this instrument reading?
Red basically means we push that frequency up. So the brain has more power, more energy for that specific frequency. It is more than normal.
Okay.
Before the machine was on, it was green, it was in the normal range. After we turn on the machines, we boost the brain power, so it went to the alpha. And the question is
And
usually,
you're measuring this by the frequency of the electrical signals coming from the brain. You're not measuring the light that you're into. Is this an indirect
yep,
consequence of the light therapy?
that's exactly a very good point, because this machine is working in the gamma frequency.
Mm-hmm.
And the frequency it's stimulating the brain, it's about 40 hertz. But what we are saying here, it is 10 hertz stimulation, 10 hertz frequency. And there is one concept
So
about
I'm
the,
sorry. Let me step back there a moment. So
yeah.
the observation in the brain is that you're getting 40 hertz signal, but the therapy that you're administering is 10 hertz. So the light is pulsing at 10 times a second.
No,
Is that correct?
actually inverse.
Oh, it's the inverse.
The
Okay.
stimulation is 40 hertz,
Okay,
but the
stimulation is
effect,
40 and the effect is 10. Sorry, I got that mixed
yeah.
up. So if I got it mixed up, I'm sure some other people did, unless
Oh,
I'm
I'm
the
sorry for
only
that.
one that's not following it. But I was confused with that. So thank you for allowing me to clarify that. So it'll
Sure.
reinforce what we're seeing, because this is not necessarily easy to follow, and it helps to
Yeah,
reinforce it from a different perspective occasionally.
yeah, and but over this 10 hertz or alpha frequency, it's not the only frequency we are targeting, because the brain network that's responsible for generating different frequency, they are all interconnected. And we have a concept in neurophysiology, we call it cross-frequency coupling. So it means if you boost or modulate one frequency, because of the interconnected network, you may modulate the other frequency as well. So if you are stimulating brain at the gamma frequency, you might also change the other frequency band, like a theta, like alpha, or the beta. And this changing or modulation could help or sometimes could stop or halt any cognitive task. And alpha is, it has been shown, it is very important frequency for the rest. And also we find it in the some mental health disorder. This alpha frequency usually is very low or very slow. So pushing the alpha up and normalizing the frequency, it's really helping for cognitive skills.
Welcome back, Dr. Lim. That was a
Thank you.
very impressive demonstration. And what I did not mention earlier is the term that's given to the type of therapy that you're administering there. And what we'd like to discuss and expand on so people can understand what we just witnessed is a term called photobiomodulation. And you can see, if you
Yes.
just break down that word, photo,
Mm-hmm.
obviously
Mm-hmm.
light, and
Mm-hmm.
then you're administering light to someone's brain through the skull. You see biomodulation, bio being biology, life, and then modulating, it's
Right.
changing it. We saw it. We saw the light impacting the skull,
Right.
changing neurological activity.
Right.
So it
Right.
was a really, we can discuss it sometimes, but I thank you for making it available. It's not, we typically do in our interviews, but it was a powerful illustration of the technology. So I think we need to backtrack now and really lay a foundation of what's going on. And what we're administering is near infrared, or what you're administering and have done a lot of work with, is near infrared light. And you can discuss those frequencies. And the mechanism, which we think works, is because this near infrared is interacting with one of the proteins in the inner mitochondrial membrane, in a part of the member of the electron transport chain, cytochrome C oxidase, sometimes
Mm-hmm.
referred to as CCO. And this particular is a chromophore, which means it
Right.
is a molecule which attracts light like a magnet and actually feeds off of that. And I just want to go on a little tangent because this is important. So, the frequency that it is in near infrared range, about 810, 830, somewhere in that
Right.
range.
Uh-huh.
So the concept I just want to explain, and I'll hand it over to you in a moment, is that most of us don't understand that when we eat food as fuel, we think that's going to nourish our cells. Well, it does, but it does it only indirectly. There is no way the macronutrients you're eating are converted they kind of leak through, you break them down in your gut, and they go into your bloodstream, and they go into your cells and feed them. That's not the way it works. It works only indirectly. It works because those molecules get broken down to essential ingredients like the fat and glucose into pyruvate, and they feed the mitochondria. The mitochondria will generate the energy, and and ultimately they're processing electrons to generate ATP. But here is the key, and I just want to finish this thought, is that that's not the only source of energy you get into your body.
Absolutely.
And
Yeah.
hardly anyone, virtually no clinicians understand this, is
Mhm.
that light is a powerful fuel for your body. And I believe, it's my serious belief, that this is one of the reasons we have an epidemic, as I referred to earlier, a tsunami, literal tsunami of Alzheimer's disease. It's going to devastate our country unless we understand this concept. But we have to fuel our body and our brain specifically with light, because as we've seen, it comes in there and it feeds our mitochondria just like food. So
Right.
for that preface, I'll hand it over to you, and you can go off, and I'll guide the conversation as we go on, because there's a lot of other concepts I want to interject here, because it's really important one. It's not one we've really talked about a lot previously on the site. It's a new evolving passion of mine, photobiology, and I'm just delighted to connect with
Mhm.
experts like yourself.
Mm. Well, you've just touched on several levels here. Okay, and so let me start with one aspect. Now, so you talk about, the processing of uh, uh, glucose and to convert it energy. That's one way the mitochondria converts the energy. Now, the other major way it does is to process oxygen,
Mm-hmm.
right? The mitochondria, the cells love oxygen. The cells love oxygen a lot more, say, for example, than cancer cells, which basically thrive on burning sugars. So that's an example. Now, what
Well,
photobiomodulation does,
let me just
yeah,
interject here, just
right,
because
right,
it's that what you said is a bit confusing, and yes, it's true. But the way that
right,
oxygen interacts here is that those electrons that get broken down from the fuel get passed through this
mhm,
electron transport chain, and they
right.
ultimately get passed off to oxygen, which accepts it and forms water, and sometimes it forms these reactive oxygen species. But it's this transfer electrons that gets this cycle rolling within those
Yeah.
mitochondrial electron transport chain to generate the ATP. But
So
that's
what
only one
what
way. That's only one way.
Yeah, that's just one way. So I was going to bring in oxygen because photobiomodulation actually does improve oxygenation to the cells. One of the ways it does is it releases nitric oxide back into the body. And nitric oxide relaxes the blood vessels, vasodilation, and it does that. So that's one way of looking at what it does. Now that is part of the mechanism that's happening within the cells. Now the really interesting thing is when you can deliver red and the infrared light to the mitochondria in the cells. Now that leads to, other than releasing nitric oxide back into the body, it then leads into synthesizing gene transcription factors that lead to the repair of cells. So it helps with cellular recovery. Now the neurons, which we are kind of focusing on right here, is just another cell with mitochondria. It processes, it can accept light in the same way and convert into synthesizing the transcription factors that lead to that.
Well,
Now,
let me just interject here for
right,
just a point of clarification so people
right,
understand this. The neurons that you're referring to are part of the brain. The brain is one of the most mitochondrial dense tissues in the body,
yeah,
along with the heart. So that's why it's a
mm,
great
mm,
organ to study for mitochondrial activity, because it's so dense.
right. So now directing light into the brain this way is actually very interesting. Um, and here you started off, talking about Alzheimer's disease. And before I get into what the evidence we found in our study, if you look at the epidemiology of dementia and Alzheimer's in terms of globally, what I found was countries that are in the northern hemisphere appears to have high risk of developing dementia and Alzheimer's. You look at the top 10 countries, They're all countries with winter, with shorter hours. So you're an advocate of being in the sun, in the
Mhm.
sunlight. I think that is really what people ought to do. But in the north, even if you go out there's not enough sun for you, right? So in a way, what we're doing here in directing light into the brain, literally we're lighting up the brain, as you can see from the computer images, have you might say somewhat similar effect as sunlight, but probably it's more targeted. It is at specific wavelengths that has an effect on the mitochondria of the neurons. So logically, that will lead to neuronal recovery if the neurons are, say, performing suboptimally or even damaged. So that's been found in vitro studies. And when before we did the study on humans, there were already at least three published studies of work done on mice and rabbits. And when they directed red and near-infrared light to the brain, they found that the biomarkers, the commonly accepted biomarkers of the amyloid beta plaques, actually are reduced. And the behavior are better, the mice were able to find a way better, so they have better sense of direction and so on. So what's we've done is we didn't invent the whole idea of this photobiomodulation can help in dementia and Alzheimer's. It's been done, but I think we are the first to complete albeit a small uh, study on humans that has given us very significant results. So when we did a head-to-head comparison with the drugs, say for example, with the information we had from the pivotal trial with Aricept, which is commonly prescribed for Alzheimer's, the data we got was 7 times greater with no side effect. So I think the key is not having a side effect. So the safety is pretty much confirmed. And I know that it's a small study. People are going to come up to me, hey, this is just actually we're reporting on 5 people on these on the devices that directed the infrared light to the brain, and all of them responded positively.
And this
So
study was published, or actually the primary investigator was Dr. Hamblin, or was it yourself?
Dr. Hamblin is a co-author. Like I'm the
Primary?
inventor. I
Okay.
provided the
You're the inventor of the device, but Dr. Hamblin
yeah, and
is he a cell biologist at Harvard, or what's his I know I'm interviewing him shortly, but I'm not
okay.
quite sure of his professional degrees.
So he's really I'm glad you're interviewing him. He's really the guy you want to speak to, because I think he kind of owned this whole knowledge on cell. He's not the only one, but he's highly cited. He's been he's presented many. He's authored more than 300 articles, a few books. So he really truly understands what's happening in the cells. So he's one of the co-authors of the study. The principal investigator was Anita Solmach. We did it mainly in Ontario here in Canada. So now I was going to go on to this. I anticipate criticism is a small study. You can't make a claim out of that. So we're going to bigger trials now. We're, this next trial that we're doing involve 226 people, is, randomized, double-blind, placebo-controlled, and basically the study will be out of my hands. It'll be led by a professor of epidemiology in University of Toronto. We'll have collaborators from Harvard, from Boston University. So it'll involve quite a number of people. And in the meantime, we're also doing a smaller study to get quicker data from about 40 people, and also rigorously controlled. So we're taking next steps to prove this out.
Okay,
But
good.
we have a lot of stories.
Yes, you
I need a lot of
do.
stories. Yeah.
And let me just emphasize a few points. As I mentioned earlier, and you highlighted, there is no effective drug for Alzheimer's. And any drug that is used or administered only treats the symptom. Nothing, just like most areas of medicine, treats the foundational cause of the illness. And I just watched a 1 hour special on PBS last night about the Alzheimer epidemic.
Right.
Uh, and it was really good. And we're going to hopefully stream that on our site and have further discussion of this, maybe even embed your interview in it. I don't know. But
Sure.
it just hit me. They said over and over again, obviously they highlighted the human components of this and the personal
Yeah.
trauma and challenge, and the fact that really there's no one, when you have Alzheimer's, you're not really mentally competent. So who's going to be an advocate for Alzheimer patients? I mean, it has to be a family member because they
Sure.
can't do it.
Definitely. Yeah.
We're crippled, mentally crippled. So it's an epidemic. We don't have a good solution. This is good. So I want to focus back now on the practical strategies. You've given some very compelling evidence. You've published work, with some leading, very well-respected researchers that this could be effective. So one of the reasons I started this site is to radically reduce the time that information like great researchers like yourself have compiled and put together, reduce the time it takes for that information to be known and understood and then transferred to the general public. So that's what I want to do with the rest of this discussion, to say, what is a take-home message? Now, you referenced earlier, and we're going to include this because you sent me a 5 or 6 page draft of an article prior to our interview, and had some really good graphs in there that was really profound that they never even addressed in this PBS special. And it's so obvious. It's just like vitamin D. The further north you go, the less exposure to the sun, the higher the risk of Alzheimer's. Why isn't this known? I'd never even realized it until you sent that to me, and I thought I followed this pretty close, and I obviously didn't. So if I missed it, almost no one gets that. So we're going to put those stats in there because it was very interesting, the tables you sent over. And we'll highlight that. So the take-home message is, live south. That's one of the reasons why I moved to Florida, because that's what you need to do. Now, it's not just living in Florida or the Middle East or, Africa. You have to be outside with a lot of sun on your skin, ideally. Now, obviously in the brain, it's going to be on your head. So, but that's the key. So that's ideal. And if you do that, you're probably not going to get Alzheimer's. But if you do, then there's targeted therapies like yourself. And I guess that's one of the things I want you to address, is the difference between the sun and some of these targeted interventions. Because, what you have uncovered is, I mean, the single best term I could describe it as profound. The implications are actually beyond profound. This simple application of light can have such a radical improvement on a disease that's going to devastate our society. So why don't you address the sun issue, and then how your version differs? And before we do that, though, I know I'm going on for a while, but most people also don't understand that over 40% of the energy in sunlight, 40%, is near infrared. So maybe there's some benefit to it.
Hey, you've got something there. So, well, knowing that, say if you live in the northern hemisphere and you're not getting enough sun, this is one really easy way to do it. There are people living in the tropics getting Alzheimer's as well. There might be other factors that affect it. But if you look at the statistics, most of the Alzheimer's are really in countries where there's not a lot of sunlight. So this is that intervention that we have here is a really simple way. And I like to make it really simple, and this is key. Now here's an interesting thing that we found in our study. We did 12 weeks of active intervention, and we have actually 4 weeks of no intervention at all after the 12 weeks. And we found that a number of people actually went straight into decline. Mm-hmm. So we had some measure of, say, modifying the disease. I don't know yet until the bigger trial, but it shows that Alzheimer's is a strongly degenerative disease. It has a whole lot of power on its own. So you got to keep doing it regularly, even with what we have. Now the key here is to make it really, really convenient and simple. You can't be going to a clinic all the rest of your life and get treated, especially if you're living far away. It costs you a lot of money. So that's the idea behind my invention, is to make it as simple as possible. You just press a button and that's it. The treatment is 20 minutes. And you can do it the rest of your life because, hey, just put it in your head. Your hands are free. You can go to bed with it. So that's really the principle behind it. Now, I think this also works because it is, it is simple and yet it is complicated. It's complicated in a mechanism. It is simple because all you do is direct the infrared light. In our case, we direct it to the nodes or the hubs of the default mode network. The default mode network is highly correlated with Alzheimer's in brain imaging that goes back to the mid 2000s. So it's pretty well established. And the key for me is to have an intranasal source because it is really close to, you might say close to the hippocampus, where it's really the key memory processing areas. But the olfactory bulb, which is really just right above the nose, has direct projections to the entorhinal cortex and the parahippocampal area, the memory areas. So if you treat that, you have direct. You're actually, you're strongly treating the hippocampus. If you look at the anatomy and the science. So that is. So now we have done this. It's really simple. Now here is how we can compare, say, a drug, the research and development, because drugs have to target a molecule. And if you look at the whole pathology of Alzheimer's and how different proteins are involved, a lot of this are now, say, focusing. Some of the phase 3 trials are really involved in, say, addressing the amyloid beta plaque side of it. There is another side which is basically based on the tau proteins that leads to the neurofibrillary tangles. But if you look at all the proteins involved, there are a lot. I don't know how many. So drug companies now, especially those who are on the amyloid beta cascade hypothesis, are going back earlier because they're not getting results from more advanced stages of Alzheimer's. So we're moving towards prodromal before the symptoms come out. But you have shown, perhaps through imaging or to be able to trace a biomarker in some way, that you have amyloid beta plaques. But that is saying that, okay, you've got Alzheimer's, you've got all these symptoms, there's no hope for you, it's too late. So what we have here is we're demonstrating in our early study, and we're actually continue to get results. People with more advanced stages are responding. It is dramatic. Perhaps one day you might have the opportunity to interview some of these people. It's going to be fun. So,
Sure.
so that is, I think that is going to be the difference. So light therapy is kind of holistic. It's natural. There's no side effect. It kind of get a brain to do its work. It does not do the work on its own. Whereas other ways, maybe trying to actually target the biochemistry. So that is, I think, that's one difference. And we are looking at ways to make it more effective. Our next study is going to be I would say, an improved version of what we've used in the earlier study, because we're introducing, as Reza demonstrated, gamma frequency, which is 40 hertz, 40 cycles per second into the brain. Now, what happens in gamma is when it is present while your brain is consolidating memory, it actually kind of minimize or prevent overactivity. They call, you might call it hyposynchrony. So there's some excitotoxins being formed, and it leads to, say the amyloid proteins are actually antibodies trying to fight it, and get into a situation where it starts developing a lot of it and developing the amyloid beta plaques. Now, the earliest studies that goes back 2 or 3 years have shown that when gamma is present, it actually avoids that, and therefore actually kind of avoid the formation of this plaques. Now, what's really interesting is MIT came with a huge, I think they did a fantastic report on this particular frequency of gamma on mouse models. And to cut it short, they found that when the mice are in an environment where the light is flickering at 40 hertz, they actually notice that the amyloid beta plaques are reduced significantly. After they do it once a day for an hour for 7 days, very significant. But here's the detail. They found that where it's reduced actually is in the visual cortex. That part of the brain that processes what you see. So this flickering has been, kind of received by your eyes and processed there. And that particular part of the brain has the amyloid beta plaques reduced. But, and then people haven't noticed is when they check the hippocampus where the memory processing area is, there's actually no change. So the message here is, if you can deliver flickering light of 40 hertz to the part of the brain where either directly or where it's not directly, where it's processing the information, you can actually reduce the biomarkers in that particular area. So that leads us to, to develop something that's more comprehensive. Instead of just being an environment where it flickers, we are directing 40 hertz to the hippocampus, like I said, effectively perhaps through the olfactory directly to the hippocampus. We are directing to the, the hubs of the default mode network. So based on this hypothesis, I have to say it's hypothesis because we haven't yet proven out in a big trial, which we intend to do. I hope, I do expect pretty good outcomes for even more advanced Alzheimer's patients where, these people are basically told you have no hope. I hope to provide some hope.
Well, good.
Yeah.
So let me just mention too, sort of a comment I just recalled as I was reviewing the PBS, remembering the PBS documentary I watched, because there's a little bit of confusion. We had talked about people living north having a higher risk of Alzheimer's disease than those living south, but the state with the highest percentage or number of Alzheimer's patients in the entire U.S. is Florida. Well, they live in the right state now, but they came there like way too late. They didn't live their life in that state and go get exposed. So I just want to clear up that confusion, but also add that there's I don't want to give a false impression that this light therapy that we're discussing here is some type of magic bullet. Is it useful? I'm absolutely convinced it is, but it's not the magic bullet. And you mentioned that there are people living in the tropics who come down with Alzheimer's disease. So there's clearly some other variables. So what I want to address is this Fat for Fuel, the book that I wrote that discusses how to get your body to burn fat as its primary fuel. And there's a lot of research that shows that once you do this, you upregulate your mitochondrial function, which is at the heart, really the foundational cause of this. And part of the process of developing the ability to burn fat as your primary fuel is something that we use called peak fasting or intermittent fasting, where you regular days of fast, maybe even longer, 24, a day, 2 days, 3 days. And when you do that, your body starts conserving protein, but it also starts breaking down protein in your body. And what is the marker for Alzheimer's disease? Beta amyloid plaque, which is protein, and tau protein is another protein. So your body starts digesting this. At least that's what Dr. Jason Fung does, who's a really strong in Canada. I think he might even be at your university, Dr. Jason Fung. So at the University of Toronto. Yeah. So it's so that's a powerful intervention. So I think there's a powerful synergy between getting the diet right and getting a light exposure. And then let's go back to the device that you put together, just because we really haven't discussed it yet, but it is infrared lighted, other than it administers it. So there's a number of different components. You have the light wavelength, which is about, is it 810 or 830?
810, yeah. 810 is near infrared, yeah.
Okay, yeah, so they're both near infrared, but 810 is the one you selected. And there's some studies that said 830 might be better, but I mean, whatever reason, you picked 810.
I can give you
So
the reasons.
okay, well, we can maybe discuss those, but that's
Right.
a little,
Okay.
that's a fine point. But it's essentially
Yeah.
it's near infrared, and it works.
Mhm.
It
Yeah.
clearly works.
Hmm.
So, and you're administering this through these LED modules. You got like 4 of them on the scalp. They're held together in this sort of metal frame break, and also you have an intranasal LED which
Yeah.
puts it back up. So you got 5
Right.
sources of LED light going,
Right.
and it's
Mhm.
pulsing currently at 10 times a second or 10 hertz.
Yeah. That's the alpha model. Yeah.
But the one that I don't think is available yet, or is it?
Yeah.
The
We're
gamma model that you're talking about is still in R&D, and that's not available yet because this is new research.
actually it's available. Actually, it's
Okay.
available like this week.
Oh,
They
okay. Okay.
just got it out. Now, what I found was, with the discussion, I can see how gamma is really going to affect Alzheimer's,
Mhm.
but there is some trace of people using gamma and getting, some slight agitation and hypersensitive to the environment. But alpha has which is 10 hertz, and
Mhm.
that was original study,
Mhm.
and it is used by a lot of people now. It has a lot of actually good effects. Now I picked 10 hertz mainly because, and Michael Hamblin can talk to you about it, because he did an animal study and looked at different pulse frequency and found that 10 hertz actually, you can interpret it as accelerating the recovery of the neurons in the animals. So his lab simulated traumatic brain injury in the mice, and then try to find out what frequency at 10 nanometers, which is what we're using here, give you the best chance of healing, quickest healing, and less. They use something called a neurological stress scores to measure the least stress on the brain, which is really at 10 hertz. So we actually have double 10 hertz, uh, mainly for its ability to, to cause or, or simulate the neurons to heal. So that's the reason for 10 hertz, and it seems to be, generally great. Now, 40 hertz is
Which is
something
gamma. That's gamma.
that's gamma. 40 hertz has been thought of, but actually, to be fair, I didn't know that it has the effect of reducing the amyloid beta biomarkers until the MIT study recently. So now we've got 40 hertz, and yet to be proven in a rigorously controlled trial, but people being treated to this are showing actually quite dramatic results. And maybe, like I say, one day you might want to interview them.
Sure. Yeah, that would be
It'd be
interesting.
fun. Yeah, yeah.
So the key here, though, is well, if you have, I'm convinced from the evidence I've seen and reviewed that if you know someone has Alzheimer's, that this is a serious problem. Obviously, it's typically pre-terminal, and here's the issue. I mean, it's going to cost us more. It's going to be the biggest cost of our healthcare, and most of the people with Alzheimer's are obviously older, and so Medicare picks up some of the cost. But what they don't pick up is the enormous cost of caring for these people, because these individuals are so impaired, especially at the later stages of the disease, that there's no way. Obviously, they can't live by themselves, but they need someone full time to take care of them. There's just no way. They will be dead. They'll leave the stove on or start a fire or something. Someone has to watch them. They have to be cared for 24/7, and that's expensive. We're talking $3, $4, $5, $6,000, not a year, every month that they have to pay to be in one of these assisted care facilities.
Right, right.
So the device you put together costs about $1,500 or so. It may seem
It's
kind of
1750 actually.
oh yeah, so it's about 1,500.
But
You're gonna
you're right.
okay, so it's less than $2,000.
Right.
No, it's a small fraction of what you would pay for a month of assisted living, and if it can provide that type of relief and give the person their life back, allow them to interact with their family and friends, I mean, that's just incredible. So there's no doubt in my mind, if I had someone close to me, fortunately my parents don't have Alzheimer's and they're close to 90 at this point. They have the preserved mental facilities, but I would be putting them on it in a heartbeat. But I don't think and I may be wrong. And when I heard your initial interview on Ben Greenfield, I said, oh, this isn't for me. I get sun exposure all the time. So for those of us who are younger, and I've actually was at presentation in West Palm Beach last week, and one of the presenters gave evidence that almost everyone over 30 has some form of Alzheimer's, although it's really mild. So all of us have it to a certain extent. But I'm wondering, I'd like you to discuss this concept. I think it's ideally, maybe everyone could use this preventively, but I don't think it's necessary. I think if you are proactive, understand that we have a tsunami of Alzheimer's coming, and you need to take aggressive, proactive preventive measurements, and the best one is to get out in the sunshine every day. Second best, I think, and this is what I want you to express, especially with the energy transfer, because what we didn't talk about is fluence, or the energy radiance and the energy density of the light being transferred, because there's a Goldilocks dose. You can get too much, you can get too little, and it just won't work. So that, and you're an expert in that, and I want you to address it. But I want to really integrate this into the recommendation that we're providing people. If you want to be proactive and preventive, that get in the sunshine, that's the best. There's no charge for it, it's free. If you want to take another step up, what I'm actually doing personally and playing with is a handheld that I can actually put a device that's a heat lamp that I just rub over my head. It's 10 watt bulb, so, and it clearly is near infrared. So it's an analog, you get the whole spectrum, not just 810, you get 810 and up to 2000, and you just spread it over the head, and I can get all, see, your device only hits certain areas, whereas if I hit this device, I can hit the whole brain. So I'm wondering if you can comment on that strategy, that the sun exposure and this infrared lulls versus as sort of an intro preventive strategy, as opposed to the neuro device that you developed.
Well, the device actually eventually reached the whole brain because you're pointing at very active hubs. So they communicate, and eventually it does that. And I think the nose is very key because of its proximity. It's really easy to reach the brain, and we know now that if you can deliver near infrared light to the brain, it does good things. So the whole thing is meant to be really convenient, but there are other methods too. You can actually direct high energy near infrared, if you like, to the brain. The principle is very similar, so it should work, so it could be a matter of convenience, but I'll say that I try to make it really simple. It runs on rechargeable battery. You just charge it like a cell phone, and it does that. So there's no one way. And here is the thing that I might want to touch on, and Mike Hamblin might probably do that too, on this biphasic response, as we call it. Now, in the field of photobiomodulation, low energy is good because studies have shown that if you can deliver just 1 joule, there's really little to the cell. Just imagine the cell is micro.
For those non-scientists or physicists among us, which is the majority, can you explain what a joule is?
A joule is actually a measurement of energy. It's a function of power and time. So you might want to have very low power and more time, because your body, like I start with 20 to 30 minutes, because this is generally the amount of time that your body needs to get into the healing process. So you can have high power and still arrive at the same joule in very short time. So I start with that. It's also good because then I can have the safety aspect of it coming in. Keep it low power and avoid, so if you have high power, there's also the possibility of getting the thermal effect. Thermal is generating heat. Now, in the law of physics, energy cannot be created or destroyed. So you have a lot of power, energy's got to go somewhere, and it does work at development of heat. But the mitochondria doesn't need heat. All it needs is the photons. So I try to keep it low.
Well, wait. Let me just back for a point of clarification. Heat, in my understanding, is infrared. So the heat that's being generated, I mean, wouldn't that part of that be infrared radiation that's transferred to the mitochondria? Or am I mistaken? Is it a flawed concept?
So photobiomodulation actually, if it's done correctly, is a non-thermal effect.
Interesting.
No heat.
Okay.
No heat, just the photons. Now you get heat mainly because the water molecules is being activated. So that generates heat. You feel comfortable. It's not a bad thing, provided it's not too hot. But it doesn't do anything in, say, synthesizing gene transcription factors. So even longer wavelengths.
But what it might do, and I think this is one of the reasons why the being exposed to the sun is so useful, is that it's, we talked about cytochrome C oxidase being energized and providing ATP, but the other function, actually, I read in Dr. Hamlin's paper, is this higher portion of near infrared over closer to 1,000 or a little bit higher,
Right.
that actually, and maybe up to 2,000, that stimulates, resonates with the water molecules, that
Right.
recharges the energy, their ability to store energy as a battery, and then transfer that energy as another source of power to the body.
Yeah, you actually have a different chromophore at the longer wavelength. Like in his paper, he studied 9080.
Yeah, 9080, right.
So that is out of cytochrome C oxidase. Here you're talking about water, water as a chromophore in the cells, receiving that, opening up the ion channels, and does pretty much similar thing. Now, what was interesting is they were wondering why in some people, the lower wavelength, like what we're using 810, is working when they do a higher wavelength. It doesn't seem to be working, but some other studies working. So it appears that water as chromophore actually activate a very, very low power. More than that, it just doesn't do anything other than, say, creating heat. So it does work. I don't know yet how far it goes before it actually stops working.
Yeah, because
at about 1,000, it does that.
Biphasic effect that you referred to, that it's not intuitive, that there's a dose that will actually improve it, and then there's another dose at a higher level of energy transfer that will actually worsen it and inhibit function.
Right. So let's go back to this and go, this has relevance to the whole history of photobiomodulation, where it was low-level laser therapy before. Now, if you think about it, this whole thing started when laser came about. And in the early days, let's say in the 60s, lasers was used to cut stuff, destructive, remove scars, shaving off materials and so on. So high power laser you can associate with this destructive. And the whole idea about low power laser actually doing the opposite virtually came out by accident by a Hungarian professor. He published his paper in '67, showing that when you shave off the fur of the back of the mice in one group and another in a control group, he found that when he directed laser at very low power, the fur on the back of the mice grew more quickly, at least double. So there's something to it. This is completely new. It's a huge discovery. Then more experiments were conducted and found that they found the therapeutic effect of low-level laser. And then later on, in the 90s, I think it was promoted by NASA. Who found that, hey, actually, you don't need laser. All you need is to deliver that wavelength into the cells, and the cells will process it, and you get the same effect.
Yeah, they also showed it wasn't just near infrared; it was the red too, like 650,
red
660.
too, exactly.
So the red and the near infrared, and probably different ranges in the near infrared are useful. There's probably three sweet spots.
Yeah. So the early days was visible red. They created a laser, through helium neon gas, which was 632.8 nanometers. So today we have 633, and some of the early research found that actually that particular part of the spectrum is, they call it the action spectrum. So there are certain part of the spectrum that actually does work, and certain part of the spectrum actually does nothing. I think you're talking about, say, 780, 790. It's that part of it is a waste of time. So where it is actually matters, but I think it doesn't explain clearly, because these are clues and indication. We found that even, say, 650, which is not identified as part of action spectrum, has been effective. So there's quite a lot of the big window.
Yeah, I want to get back to the preventive recommendations I asked on the question initially, and kind of hold you to the fire, and so that you can answer that. especially when you just commented that, there appears to be sections of the spectrum, and when we say the 780, 630, 810, we're talking about nanometers. These are the wave,
Right.
the nanometers, the wavelength of the light. You referenced that there's sections that appear not to have any biological benefit, and that kind of reminds me of the early days of the Human Genome Project, where they thought there's these massive sections that were junk DNA. We don't need these, and now we're finding this more important than the regular genes. So it's just because we didn't know, and I think we may be too early at this stage. And I tend to refer back to a lot of the pioneers in this that have done this work 100 years ago, when we didn't know even a fraction of what we know now. But that they had an analog approach. There's the whole spectrum, and we acknowledge that people living up north, down south, have a relative resistance because they're getting exposed to the full spectrum, which clearly has a near infrared, but it has all these other frequencies. There might be some other benefit we have yet to figure out.
Yeah.
So, and actually, one of the benefits for the infrared that you said was growing hair in the mice, but they actually have LED devices that you can buy now on Amazon.
Red, actually,
yeah, the red. I'm sorry, you're red. Yeah, to grow your hair, but there can be 100, a few hundred, or even more. And this device I referred to earlier is only $27, and you can just rub it over your head and get the same benefit. Now, it's not an LED; it's a heat lamp, so it's an analog, and but it's a lot less expensive. And so, comment on that as a preventive strategy, and also the sun, and help us understand the difference, because it has to do with this energy density, this joules per square centimeter of energy that's transferred, which may be too much, and it gets to this biphasic approach with if you have too high a dose versus too low a dose and be targeting. So it's a complex process to understand, but I think it's important if one seeks to apply this information to themselves personally.
So first and foremost, I think the sun is great, probably the best if you go into the sun, as long as you don't get overexposed to UV.
Right.
Yeah. So maybe 20 minutes a day in bright sun, something like that. I think that's really the most natural.
It depends on the season. In some cases, it's skin color, the latitude, the altitude. So for some people, it might be 3 or 4 minutes. For others, it might be 4 hours.
Absolutely.
So it really ranges quite widely.
So that's probably a great way. The lamps, I tend to, I tend to put safety first, so I try to keep it, as low power as possible, as long as it activates what it does. And when you have near infrared, when it penetrates quite deeply, you don't actually need a lot of power.
How deeply would you say, from your experience?
Well, you just saw the
from the EEG. So what have you been able to correlate? We're looking at 5 centimeters, 6 centimeters, 8. How deep are you going?
So the only way you can measure with a light meter is in a cadaver,
which is not. So that's not a biological system.
Yeah, that's not a real thing. So people have measured. It goes down to 5 centimeters, and it's depending on the power. If you use laser, it probably goes actually more deeply. If it's near infrared, actually, experiments have found that 8-10 nanometers actually go the deepest in the live, especially in a live environment. And why is that? It's because that as you go beyond 8-10, it gets absorbed by water.
Right?
More and more. So the absorption is actually exponential.
That's right. So because 810 doesn't power water, it powers the mitochondria, and the cytochrome deoxidase specifically, and but it's the marriage of those who's that powerful synergy where you're getting this intercellular storage in your battery and recharging the mitochondria. It's just like so crazy, such a simple concept, and the more I study medicine and health, the more you realize the simpler it is. But there's a lot of complexities to it, and thank God there's researchers like you to help us figure it out.
Yeah, well, there's a community of us. Actually, I'm not the only one, but I think some of the work that's been done before me has been really, really useful. I got to give credit to a lot of these people, but we're still discovering. I don't think we're anywhere near what we need to know, and I'm doing my part in trying to reach perhaps into customized medicine, especially with our, I think our research in integrating with EEG is cutting edge. The more data we have, the more we're able to identify profiles with different levels, even like I said before, Alzheimer's, different levels of Alzheimer's, and how we're going to do that. And there are other conditions as well. If you have traumatic brain injury,
Mm-hmm.
so research is done in, say, particularly Margaret Naser of Boston University. She's done more than 10 years of work in looking at the effects of photobiomodulation in the brain. She's one of the co-authors, by the way, in our last study. So it works. Now, the only thing we need to do is to put all that into much larger clinical trials. That's pretty
Sure.
expensive.
Well, my
We can
guess is
afford,
it would also work for all the neurodegenerative diseases, like not only
theoretically,
Alzheimer's
yes.
but ALS and Parkinson's. And Parkinson's is not as common as Alzheimer's, but it's sure devastating.
Oh, yeah. So I think Parkinson's, in my opinion, is more complicated to treat from photobiomodulation, because if you direct in certain ways, it's not, say, reaching the substantia nigra or maybe the basal ganglia and so on. But we have, we've seen Parkinson's patients responding, but it is much more varied. So I think what we need to do is to point the infrared from the back here at the below the occipital area called the occipital protuberance, nearer to the substantia nigra. So we're actually working on that. So let's see how that pans out. I think theoretically that is going to help.
Okay, good. So
yeah.
I want to give people strong, concrete, clear recommendations. So before we go, I want to beat this thing again. So to let people know, it's not just a light, that's not a magic bullet. It's an important part of it, a vastly underappreciated one. But one of the keys is to get your body to burn fat as a primary fuel, because that will fuel your mitochondria, the optimist. And ultimately, Alzheimer's is a mitochondrial dysfunctional disease. And if you've got impaired mitochondria, you're not going to. That's the reason why this infrared works. It's charging up your mitochondria, cytochrome C oxidase specifically. Now, if it didn't work that way, you wouldn't see the results that we demonstrated earlier. So the practical thing, I think we're all in agreement. If you have advanced Alzheimer's, it's a serious problem, then the targeted therapy that you develop is great. Now, you also have an intermediate device that's just the nasal probes only. So I'm assuming that's less. And then the two steps below that, preventively, would it be fair to summarize that to be preventive, get out in the sun as much as you can? If you can't, for whatever reason, and you don't want to spend the extra money because your finances just don't support that, then go to Amazon, get a $27 heat lamp, and rub your head every night before you go to bed. And just to comfort, and don't go crazy, but put it over your whole skull. So would you say that's a fair summary of your years of research and what you've learned?
Yeah, when you use something like 10 watts, keep moving it.
Mm-hmm. Oh, yeah.
It's quite a bit of power. Yeah.
Well, the device I recommend has two settings. So I imagine one is like half power. So it's 5 watts. And the 5 watts, pretty much you keep on almost indefinitely at one spot and not be uncomfortable. But at 10 watts, you're right, you do have to move it around, otherwise you will get some discomfort. But let your body guide you. Always listen to your body, get the feedback, and you won't run into troubles. But it's still a lot. You want to use it. If you use a 250 watt light bulb, but you get into a lot more challenges, because with the heat lamps, only 12% of the energy is near infrared. The rest of it is mid infrared, which is transverse heat.
Let me give you a clue. The moment you feel heat, just move it.
Yeah,
really,
right.
You don't want heat, actually, in photobiomodulation.
Yeah, that is a very good clue.
Yeah.
And just to let me finish this, that device I'm referring to is not for photobiomodulation. It's sold as a device to help muscle pains. That's what it's used for.
It help, yeah.
Okay.
Actually, heat helps muscle pain. Yeah.
Yeah. So would you say that it might be wise to go up a little bit higher if you have a muscle injury you're trying to treat? Or it's the same thing. The moment you feel the heat, move it.
Yeah, I know one thing I believe is, actually these are all pretty well researched. When you have muscle pain or pain in certain parts of your body, if you can direct low level laser in that focal area, we have tested in our devices too. It is quite remarkable. You get you know, things like, even systemic pain. But if you have like, carpal tunnel syndrome or tennis elbow, you put low-level laser, you'll find that you very often within half an hour the pain goes away. And if you go to chiropractic offices, a lot of them have this clinical devices with low-level laser.
Yeah.
And here's the interesting thing that I found. Um, even in the tropics, people get a sun, but they still have pain.
Mm-hmm.
So there is something to say about directing something more focal in
Mm-hmm.
particular areas of the pain, where you direct basically a monochromatic waveline that affects the mitochondria, and that seems to work even where you have a lot of sunlight. So in a way, it's complementary. If you're in the sun often, you're probably healthy and you're probably tough as
Mm-hmm.
hell. But sometimes you need some help.
No, there's no question. But then you have this whole biphasic issue. So, like, I've got
Yeah,
a 15-watt laser that's typically used in many chiropractors' office called the K laser.
yeah, right.
But I've been having second thoughts about it and understanding this biphasic approach because it may be too much power. And it's most of the research was done with low-level light therapy and very low power. So, if you get too much, it's going to actually inhibit the improvement rather than stimulate the improvement.
Right. Yeah, yeah. Yeah, so no one knows exactly where it starts to, have the opposite effect. It all depends on the body,
Mm-hmm.
but it's really it varies from person to person. But generally, if you can deliver low energy, just enough for the cells to respond, you don't need to go higher than that. That's
Yeah.
my personal opinion.
Yeah, I love that. And actually, when I learned of your work and started reading your studies and information that you were publishing, it really catalyzed my thought in this process and really reshaped it to the appreciation that you don't, just what you just said, you don't have to be high. I mean, you don't have to go in there beat it with sledgehammers. You can just like
Right, exactly.
massage it and tickle it and push it a little bit, and that's all it needs the push it needs to improve and stimulate its progress towards healing and health.
Yeah.
All right, so this has been fantastic. We are going to have Dr. Hamblin on, he's the deep researcher. I've been doing this for a long time at Harvard, and he will certainly complement the information that you shared with us today. But I am deeply appreciative for the time, effort, and energy you've put together to provide a commercial device that's available for this devastating neurological illness that can help so many people. And really, it may seem like a lot, but for someone with this disease, it could radically reduce the total cost because we're looking at tens of thousands, in many cases, $100,000 a year in extra expenses that is not covered by insurance. It has to come out of someone's pocket, and frequently the pocket runs dry pretty quickly because that's a big expense for most people. And then they're left with this psycho-emotional trauma of not being able to care for their parents who cared for them, and it's just too expensive. They don't have enough resources to do it. So this is a simple one. Combine it with the Fat for Fuel, which is coming out in May, and I think we've got a powerful synergy that can go a lot to really hit this epidemic of chronic disease and affecting the brain.
I just want to add that what we have is considered low risk general wellness device. So we don't actually, we don't make particular claim except that we've just done this study on Alzheimer's, so we can point to some of the evidence. But for me, safety first, and it helps
It's
with
low,
mental
it's low
acuity.
level. It's a lot safer than
Low
a
risk.
15-watt laser, of course. And there's virtually no risk. I mean, there might be something that we figure out. Maybe you get a little agitated or something, but essentially it's just simple light. So
Right.
the toxicity risk is incredibly low. I mean, it's safer than any medication. There's no question about it. I mean, even safer than water. Water can kill you. So excess water can kill you. So
Right,
like
yeah.
any. All right. Well, thank you again. Appreciate all your effort, and we will look forward to upcoming reports from you in the future.
Well, thank you for inviting me. It's been a pleasure.